Hello students, this video is about cellular membranes. So cellular membranes are one of those structures that are really conserved. They are present in all living cells. For that reason, we're going to spend a little bit more time getting deeper into their structure and their function. First, let's review the function of it. So we call it the cellular membrane, sometimes the plasma membrane. It's going to be important in separating living cells from the outside environment. It's also going to do a little work with protecting the cell. So one thing that all living things need is homeostasis, or some sort of balance within the cell. For that reason, cell membranes are selectively permeable. Permeable means going in and out. So some substances can come in, some substances can go out, but not all of them can. So the primary role of the cellular membrane is allowing certain things in and certain things out, but not everything. Down here at the bottom, we see cellular membrane present in an animal cell, in a plant cell, in a prokaryotic cell. We see it again here. Cell membranes are something present in all living cells. Okay, let's go over the structure. So there's a couple of different things we are going to see. Phospholipids, proteins, cholesterol, and some carbohydrates. Let's go into each one of those. First, the phospholipids. So remember back to our macromolecules section, phospholipids have one part of them that's hydrophilic, meaning it likes to be by water because there are some polar bonds in here, and then they have hydrophobic tails. These tails are just a bunch of carbon and hydrogen atoms bonded together. For that reason, they are non-polar. Because they are non-polar, they're hydrophobic. They do not like to be next to water. For that reason, when they come together, we're going to have the hydrophilic side facing the watery, either like outside the cell or inside of the cell, and then those hydrophobic tails hanging out together. Notice those tails are trying to avoid water, those heads are trying to be next to water. That's going to create the structure of the membrane. These are not actually bonded together at all, they're just hanging out together because of those forces about whether or not they want to be next to water or away from water. Because of that, we get our phospholipid bilayer. Bi means two, so we have 1, 2 layers coming together. So we don't have these as sort of a static thing as we normally think of them. Instead, there's going to be some movement allowed with them. So sometimes we see this called the fluid mosaic model. This is going to be where the membrane is able to move a little bit, and then these proteins and these glycolipids and some of the other stuff that we see is going to be sort of floating around in our model. So let's look at those things that are floating in a little bit more detail, figure out what they are. So the first thing we have is a glycoprotein and a glycolipid. So this prefix glyco refers to carbohydrates. So a glycoprotein is a carbohydrate bound to a protein, and a glycolipid is a carbohydrate bound to a lipid. There's many glycoproteins and glycolipids that are going to be on the surface of the cell. They're going to be important in recognizing other cells or recognizing self versus non-self. They're also important in transporting things between different cells, in the structure between cells, etc. Down here, there's a little bit of an image of what it might look like. For example, here's a protein. On the end of the protein, there is a glycoprotein, or there's carbohydrate bit right here. That's going to allow things like recognizing toxins, recognizing viruses, recognizing bacteria, etc. There's also going to be cholesterol present in our cellular membrane. So the role of cholesterol is to allow just enough movement for the right amount of fluidity in the membrane. So what it's going to do is it's going to add a little bit of space between the fatty acid tails. So when fatty acid tails are really straight, they're able to come really close together. Because those tails are so close together, there's a type of interaction called van der Waals forces or London dispersion force, where they're going to induce just a little bit of charge, and they're going to stick together a tiny bit. So that's a really weak bond, but it's enough that if you have these lipids all really close together, you're going to have a solid membrane, which is not what you want. Instead, we want a fluid membrane. Cholesterol is going to go in there and add a little bit more space. We see this in organisms that live in cold environments and organisms that live in warm environments. So if something is cold, there's naturally less molecular movement, and so it's more likely to form that bond. So cold environments are going to need a lot more cholesterol to push apart the membrane to allow the right amount of fluidity. versus warm environments where there's naturally more molecular movement, they're going to need less cholesterol in their cell membranes. We see down here our phospholipid bilayer and some cholesterol pieces, this modified steroid stuck in there to allow just the right amount of fluidity. So here's a couple images just showing what we've talked about so far. So we have our phospholipid bilayer with the cholesterol in it, and then we either have glycoproteins, glycolipids, that's going to be important in cell recognition and cell communication, etc. Notice in plant cells, when we have our cell wall, there's not all those glycoproteins and glycolipids. Those are mostly just in animal cells. Okay, so now let's talk about some things that can get across the membrane. So we call this transport. So first, let's just review the idea of diffusion. So things naturally would like to move from an area of high concentration, where there's a lot of them, to an area of low concentration. That happens naturally. It does not require energy in order to do so. So one thing that the cell membrane is going to allow is certain things in and out using diffusion, but it only allows certain things in and out. So it has this selective ability. There's going to be active transport, which is going to require energy. There's passive transport, which is based on diffusion, and that's going to allow certain things in and out. So let's first break down passive transport. So there's two types. First, there's simple diffusion. A very few number of really, really small, non-polar, non-charged molecules are able to travel directly through the membrane. A really good example is oxygen and carbon dioxide. So if you take a cell that's low on oxygen and put it up next to a capillary that's really, really oxygen-rich, then those little tiny oxygen molecules are able to go right through the membrane into the cell. Remember, oxygen is O2 in its molecular form, and that is nonpolar, very small, so it can go directly through the membrane. Most things, though, cannot go directly through the membrane. So instead, they're going to get some help from some proteins. So sometimes we call these transmembrane proteins, meaning they go across. Sometimes we call them pores. Sometimes they are helper proteins. But these proteins are going to allow certain things through the membrane. That's what this image here is showing. Notice we have a high concentration and a low concentration. So these things are moving with their gradient, but they need this protein to allow them through the membrane. That is called facilitated diffusion. Okay, then we have osmosis. So osmosis just means the movement of water. And sometimes when certain things cannot move through a membrane, water will move through in order to equalize it. So remember, water is polar, so water cannot move directly through the membrane. Instead, it's going to move through a type of protein called an aquaporin. But water wants to equalize things. What that might mean is if you have a cell that has many solutes inside of it, and there's water on the outside, the water may want to come in to the cell in order to equalize that solute concentration. The terms we use for that is hypertonic, refers to the side with more solute. Remember, hyper means more, like, oh, you're so hyperactive, means you have more energy. Hypotonic refers to the side with less solute, and then isotonic is when they're equal. That's going to allow some differences to happen. So if we see up here in this image, we have a whole bunch of solutes represented with the yellow, but they can't get through the membrane. Instead, water, represented with the blue, is going to move from where there's more water to where there's less water in order to equalize the concentration. Sometimes that can allow things like this, where we have a membrane here, and the water is going to flow in this direction to try to equalize. Let's look at a couple different examples. If you have a red blood cell has a certain number of solutes inside it. If you put it into a solution that's hypertonic, meaning there's more solute, then the water is going to leave the cell in order to equalize it, and the cell will shrivel. If you put it into a hypotonic, meaning less solute, water is going to enter the cell, sometimes causing that cell to burst or lyse. If it's isotonic, meaning it has the same amount of solute inside the cell and outside, water is just going to move back and forth, and there's going to be no change in the cell itself. So all of those things are happening passively, whether it's like straight diffusion, whether it's facilitated diffusion, whether it's osmosis. But a lot of times, cells want to move things against their concentration gradient. and that requires some ATP energy. So here's an example where we have a lot of things inside the cell, but this wants to continue moving them inside the cell against its concentration gradient, so some energy is required. This example right here is one that we're going to focus on a little bit. This is a potassium-sodium pump. One thing that we notice is there's the inside of the cell and the outside of the cell, and the potassiums are going to be moving to the inside of the cell. The sodiums will be moving to the outside of the cell against their concentration gradient. And so we're going to end up with a lot of potassiums inside the cell, a lot of sodiums outside the cell. For every 2 potassiums that are moved inside the cell, 3 sodiums are moved outside the cell. They both have a +1 charge, and so we're going to end up with a more positive charge on the outside versus the inside, and so we get a little bit of a negative charge inside the cell. This is important for this reason. The pump creates this electrochemical gradient between the 2 sides of the membrane, and then you can use that gradient to accomplish cellular tasks. Specifically, this is how your nerve cells send signals. So this is what it would look like. We're doing this to allow these differences to be created. When these differences are created on the inside and the outside of the cell, then that is called its resting state. Then if you go and poke that cell, it's going to try to transmit some sort of signal to the rest of the cell. And the way it does that is it's going to open up some channels to allow these things to go back towards their concentration gradient. That's going to cause a difference in the electrochemical gradient that is able to go along the length of the cellular membrane, then eventually get turned into a chemical gradient and allow these things to communicate. So the way that your nerve cells communicate is using this process. Here's another example where we have this movement along here, where we have these things that are naturally set up to be negative and positive, and then as a gradient gets restored, then a signal gets transferred. So if we were to break that down into the different steps, we would say a sodium-potassium pump uses ATP to pump more sodium ions outside the cell than inside, and more potassium ions inside the cell than outside. That's going to create this little bit of negative charge. This is millivolts, just a very small negative charge on the inside of the cell relative to the outside. Then, when there's a disturbance in the cell, the membrane is going to open the sodium channels, causing that little bit of negative to go away. We call that depolarization. Sodium ion channels are then going to shut, and the potassium ion channels are going to open. Then the sodium-potassium pump is going to restore that polarity. As this goes along the length of a neuron, that's how your nerves communicate messages in the body. I think it's pretty cool. So the last thing I want to talk about is how large amounts of things get either brought into the cell or brought out of the cell. It's called endocytosis and exocytosis. So endocytosis means bringing in, and exo, like exit, cytosis means going out. They're processes that allow larger molecules to move across the plasma membrane. So instead of going through a protein, instead what's going to happen is an entire chunk of membrane is going to either fuse with the overall membrane or pinch off to go inside. Here's an image of that. Let's say we're trying to get these molecules inside of a cell here. They may come and they may form a little divot inside the membrane. Eventually that'll pinch off to form a vesicle that's now inside of the cell. Same sort of thing with exocytosis. There's a vesicle, just a little storage container filled with whatever it is. As that goes against the membrane, it's going to fuse with that membrane, and as the membrane pinches apart, this thing becomes part of the membrane, and whatever it's carrying is released. So membrane that encloses a particle as a fluidated membrane allows this movement, and this is going to be how things are transported either in or out. Sometimes we call it pinocytosis if we're bringing in fluids, and phagocytosis if we're bringing in larger particles, but those are just some details. So I hope this helps in getting you thinking about some of the patterns that we see in cellular membranes and just how awesome their structure and their function are.